Hello, good afternoon. Welcome again to the third webinar of this series we started at CCAM in the context of COVID, challenges and responses in simulation modeling and beyond. Today, we have two outstanding speakers, Andrea Cavalli from the Italian Institute of Technology and University of Bologna, and Modesto Orozco from the Institute of Institute of Research in Biomedics, and it's also associated to the BSc Research Program in Computational Biology. We will hear today two presentations about how we can make use of computational approaches in a multidisciplinary fashion, combined with the use of big data and machine learning to really face the challenge of understanding the structure of biomolecules and the interaction of COVID in our organisms. I remind you, as we have done in previous weeks, that we will have the presentations first. We will start today with the presentation by Andrea. After that, we will have a small break, and we will continue with the presentation by Modesto. And after these two presentations, we will have a common session of questions you can address questions to specifically to any of the two speakers, or you can also pose more general questions to be addressed by the two of them. Both Sarah Bonella here, the Deputy Director, and myself, we will be sharing. We will take care of collecting these questions and arranging the corresponding question session at the end. I remind you that you can send those questions by using the the chat possibility that you have already on the YouTube. If you have problems using that, you can also send an email to director@ccam.epfl, and we will collect them, as I said, to pose the questions at the end of today's webinar. So I hope you will enjoy this third session. So without further delay, I will then now pass the word to Andrea Cavalli, who will start with this first presentation of today's afternoon. Andrea, whenever you want.
Thank you very much, Ignacio. Good afternoon, everyone. Okay, thank you very much for this very nice initiative and the kind invitation. Today, I would like to discuss with you about current therapeutic option for COVID-19 and how how and others try to utilize computational methods, including atomistic simulation and machine learning, to try to explain and possibly also to predict mechanism of action of available therapies for this outbreak. This is the overview of my presentation. There will be a very quick introduction to SARS-CoV-2 and COVID-19, with particular focus on the stages of the disease and the molecular basis for different therapeutic options. And then I will move to computational methods for drug discovery, with particular focus on drug repurposing, also with some few words on machine learning for the same objective. And finally, if I have time, I would like to discuss with you the mechanism of action of two of the golden standard now for trying to fight COVID-19, Remdesevir and Chloroquine. Everyone knows pretty well that in December 2019, some strange cases of pneumonias were reported in China. And soon after, it was discovered that the causative agent was a coronavirus, in particular the severe acute respiratory syndrome coronavirus number 2. Very likely, there are many hypotheses about the origin of this coronavirus, but the most probable hypothesis that if it is of animal origin, and actually SARS-CoV-2 is quite closely related to a bat coronavirus, RATG13. Actually, the sequence identity is more than 96% with this coronavirus as far as the RNA is concerned, whereas the identity with SARS-CoV-1, the 2003 outbreak, is just a little bit more than 80%. As of today, more than 3.5 million cases have been reported worldwide, causing over 250,000 deaths. Concerning the virus, as mentioned, it is the causative agent of COVID-19 is a coronavirus, and it is a positive sense single-stranded RNA, a pretty large virus concerning as far as the genetic material is concerned, because the RNA is of about 30,000 bases. And also the viral particle is pretty large with a diameter of about 200 nanometers. Interesting enough, the initial R0 reproduction number was between 1.4 and 3.9, meaning that one single person could infect up to 4 different persons, 4 different people. It is very important from the drug discovery and drug purposing standpoint to consider the different stages of the disease, because the different stages require different targets. Very recently, 3 different stages have been identified for COVID-19, probably 4 different stages. Stage 1, in which there is the early infection, and stage 2A, in which still the viral response is more important than the host-inflammatory phase, and then the 2B and 3, in which very likely is the response of the human body more relevant than the viral infection per se. And therefore, targets for stage 1 and stage 2A should be looked for at the viral particle, whereas drug targets for stage 2B and stage 3 should be identified on the human cell, on human tissues. For example, drug targets for stages 1 and 2A could be considered the structural and non-structural proteins of SARS-CoV-2. Among the structural proteins, we may recall the spike, which is one probably of the most relevant drug targets so far identified for discovering new antiviral compounds. Among the non-structural proteins, which are 16, we may recall the proteases, the exoribonuclease, and very recently the RNA-dependent RNase, among the others. For stages 2B and 3, instead, where the systemic inflammation takes over the viral stage, the most important targets so far identified are 2 receptors for interleukin 6 and 1, JAK1 and 2 protein kinases, and some proteins responsible for the systemic vasculites, which has been reported as one of the most dangerous phenotypic and phenopathologic aspect of stage 3 of the COVID-19 disease. Talking about computational methods for identifying new potential therapy, The most obvious approach is the so-called drug repurposing, namely to try to identify among the FDA and EMA approved drugs molecules that can be immediately exploited and utilized in clinical settings. Of course, drug discovery and classic drug discovery can in a way be carried out, also discovering completely new compounds, but we should obviously keep in mind that a drug discovery and development program can take up to 10 years with an expense, with a cost, which can overcome easily $1.2, $1.3 billion. For the emergency, therefore, probably drug repurposing is the only option available to try to fight COVID-19. Drug repurposing is composed of different steps, briefly are reported, in which there is a selection of targets and database, and then there is a selection of the most suited computational approach for the identification and prioritization of drug for immediate clinical trials and exploitation in hospital and in clinical settings. For the selection of the database, again, there are many possibilities. Of course, you may consider also much larger database of million or even 100 million of compounds, considering virtual molecules. But again, the most pragmatic, in our opinion, the most pragmatic way to run drug repurposing is to select drug already available. And this is why the database on which we focus was the drug bank. The latest release include up to 2.6 thousand approved small molecule drugs, along with about 1,400 approved biological compounds. The second step is the target selection. Also, target selection is very critical because of two main reasons. Reason number one is that to have a good target, you need a good structure, a good experimental structure, or at least a very, very high quality homology model. But frankly, I would prefer to have crystallographic and experimental structure to run drug repurposing for clinical trials. And despite there are 20 proteins overall in the virus, 4 structural and 16 non-structural proteins, the only available for structure-based and pharmacophore-based exercise are these 3 proteins: the 3C-like protease, the exoribonuclease, and the spike. Very recently, it has been reported the cryo-EM structure of the RNA-dependent RNA polymerase, but in this case, the resolution of the cryo-EM structure is quite low, only 2.5, 2.8 angstrom, and therefore exploitation of low-quality target for drug repurposing could be rather unwise, particularly because the relationship, we know pretty well that the relationship between the binding free energy and the affinity constant is logarithmic, and therefore even a small error in the free energy prediction can be responsible for a huge error in the affinity constant estimation. The same holds also for the spike protein. Actually, the spike protein, the crystal structure of spike is available, but again, the resolution is not particularly high, it's 2.45. And moreover, the interaction between the RBD domain of SARS-CoV-2 and the ACE2 receptor, which is the endogenous human receptor responsible for binding of the virus on the surface of human cells, is rather shallow, you may appreciate here that the interaction region is rather shallow and not particularly suitable for small molecule drugs. Probably for monoclonal antibodies, this interface could be good targets, but not particularly suited for small organic molecules. Then we started our drug repurposing activity, combining together pharmacophore-based and structure-based drug design. Pharmacophore-based was run in a rather peculiar way because we combine pharmacophore modeling with enhanced sampling in a way that we try to refine the structure on which to run the pharmacophore analysis using enhanced sampling simulation in a very, very simple mathematical formalism. A few hundred of nanoseconds of simulation were run to equilibrate the system, and then we ran a classic pharmacophore modeling using the available snapshot coming from different, from the various nanoseconds of simulations. For example, we were able to prioritize, among others, Remdesevir, which is an historical HIV protease inhibitor, which was fitting pretty well onto the pharmacophoric model that we derived through our enhanced sampling and these simulations. Interestingly enough, Remdesevir was also prioritized using conventional structure-based drug repurposing using the crystallographic structure of the 3C-like protease and running structure-based virtual screening using the drug bank database. Remdesevir was #1 in our simulation, was ranked #1, much better than other potential drugs that I will mention later in my presentation. Remdesevir was also superimposing pretty well the binding conformation as obtained by virtual screening, was also superimposing pretty well onto the PRD endogenous ligand, which was co-crystallized into the 3C-like protease reported in the protein data bank. So the binding mode was rather intriguing and quite promising. Vice versa, two very famous drugs for the treatment of HIV, which are binders of HIV protease, were not really able to bind into the 3C-like protease binding pocket. The two molecules I'm talking about are ritonavir and lopinavir, which in combination, they are the principal, the active principles of Kaletra, which is one of the drugs most widely exploited for the treatment of COVID-19, with rather controversial outcomes. Kaletra was probably the first drug utilized in many European hospitals, end of February, beginning of March, but the clinical outcomes was rather poor using this drug. To finally confirm our prediction, was more than 1,000 compounds. I'm just mentioning about very few examples of our simulations. We use molecular dynamics for binding kinetics. A few years ago, about 5 years ago, we developed a new methodology based on enhanced sampling for residence time prediction, which is actually the active code of Beaky life science software, and which has been utilized quite extensively by many pharmaceutical companies for binding kinetics estimation. The methodology is pretty simple. It's based on enhanced sampling. Is based on scale MD, and the protocol that we devised many years ago is to simulate a certain number of unbinding events in the scale ensemble using some restraints to avoid, for example, protein unfolding. And then to compute the residence time and to compare the residence time with the experimental one, or to use the residence time as a further figure of merit to prioritize compounds in a structure-based virtual screening campaign. And the only assumption is that the entropy difference among within a congeneric series of compounds is rather similar. Here we have a very quick movie of how ScaleMD works. Here we have the crystal structure of HSP70 co-crystallized with a nanomolar inhibitor. After a few tenths of nanoseconds of ScaleMD simulation, the ligand is able to unbind the protein, and what we can calculate is the scaled residence time and compare the scaled residence time with the experimental kinetic measurements. And of course, the simulation is, for sake of clarity, we have just removed the solvent here because the movie is much more, much clearer. We ran our residence time prediction with Untitled and Lopinavir, and even though these results are quite preliminary, we could see that Untitled has a slightly longer residence time compared to Lopinavir, and seems in this way to have an advantage over Lopinavir in binding the 3C-like protease of SARS-CoV-2. After our simulations were completed, the outcome of a very important clinical trial was published in the New England Journal of Medicine. The trial combining Lopinavir and Ritonavir in adults with severe COVID-19. You may recall that from our simulation, both Lopinavir and Ritonavir don't bind particularly well into the binding pocket of the SARS-CoV-2 structure enzyme, and the conclusion was that no benefit was observed with Lopinavir and Ritonavir treatment over the standard of care. So Kaletra, which has been one of the drugs most widely utilized for the treatment of COVID-19 infection, was not particularly helpful in severe case of COVID-19. We should, however, to be completely clear, we should recall the 3 stages of the disease mentioned in the beginning of my presentation. If we are already in the hyperinflammation phase, probably any antiviral compound will be particularly helpful in recovered patients from severe COVID-19 infections. From our simulation instead, Untitled was probably the best suited candidate for binding into the protease, even though Untitled is not a really good drug. Untitled was approved for HIV infection more than 20 years ago and was abandoned basically soon after because of severe side effects. More interestingly is Fosmantinib, which is a prodrug of Untitled because of this phosphoester bond, and which is promptly hydrolyzed in vivo, and which leads to Untitled after oral administration. Other activities were also done with the other interesting target, which is NSP14, also better known as exoribonuclease, which is considered a kind of Achilles of SARS-CoV-2. Probably you recall that I mentioned in the beginning of my presentation that the RNA of SARS-CoV-2 is particularly long. And so the proofreading mechanism is particularly important for the replication of the virus. And NSP14 or exoribonuclease is one of the key enzymes involved in the proofreading mechanism after the replication of the virus. And if we are able to block this enzyme, since the replication of such a long RNA is error-prone, we can have many errors in the viral replication with the final reduction of viral infections. In the second part of my presentation, I will briefly mention about the use of artificial intelligence and machine learning for COVID-19. Frankly speaking, we use machine learning pretty extensively to analyze our simulations. So I even didn't mention about that, because nowadays machine learning is a kind of standard tool for analyzing MD and enhanced sampling trajectories, or even docking outcomes. But for COVID-19 specifically, machine learning can play an important role in, for example, in diagnosis, for instance, for a kind of automatic radiography analysis, for epidemics and epidemiology. But I would argue, and again, I didn't find many applications in the literature, I would argue about the use of machine learning for drug repurposing and drug discovery. Because machine learning needs to learn, and to learn, machine learning needs data. And if data are not available because the disease is completely new, to run drug repurposing using machine learning could be particularly complex. What we discovered a few months ago was this interesting paper published in Lancet, in which machine learning was used for literature mining, for natural language processing. And through this algorithm developed by Benevolent AI, a company based, if I remember well, in Oxford, and mining the literature, they were able to identify one very interesting compound, which is baricitinib, which was far from being trivially identifiable through other more conventional drug repurposing approaches. Baricitinib is JAK1, JAK2 inhibitor, but they also pointed to baricitinib as an inhibitor of AAK1 kinase, which is one of the kinases responsible for the clathrin-mediated endocytosis mechanism, which is key for the viral entrance into the human cell. This compound is also particularly interesting because could be one drug usable for phase 2b and phase 3. As mentioned before, the targets for these two phases should be selected among human targets rather than among viral proteins, because in this phase we have an hyperinflammation and what it has been called the cytokine storm, in which a cytokine receptor, for example, the receptors for interleukin 1 and interleukin 6, can be particularly relevant for mediating the hyperinflammation responsible for the most dangerous pathological effects of COVID-19. And many drugs have been tried in these phases. For example, tocilizumab, canakinumab, which are monoclonal antibody able to bind to the interleukin 1 and interleukin 6 receptor. But as you may see here, one of the proteins responsible for the mediation of the message through the cytokine receptor is JAK2 and even JAK1. Therefore, JAK1 and JAK2 inhibitors could be particularly suited for blocking the hyperinflammation triggered by SARS-CoV-2 in human bodies. In the very last part of my presentation, also connected to this intriguing mechanism of action and the two different phases, the viral and the hyperinflammation, I would like to focus on two molecules on which we have been working quite extensively over the last few months, Remdesivir and Chloroquine, which are a kind of golden boys, I would say, in the field of SARS-CoV-2 treatments. In particular, Remdesivir has the following structures: is a compound with a kind of nucleobase and two ester bonds, which make Remdesivir quite susceptible to hydrolyzation. This is why it is only available upon injection, Remdesivir. In particular, it's a prodrug, and the hydrolysis can occur at this phosphoester bond and releasing this part of the drug, which is the one acting as analog, as a false substrate for the RNA replication. 4 days ago, made the cryo-EM structure of Remdesivir in complex with the RNA-dependent RNA polymerase was reported in Science, published by this Chinese consortium. And was particularly interesting, this crystal structure, because basically overcome all the simulation done over the last few months by many different groups using Remdesivir. And the mechanism of action was particularly clear, not only looking at this picture and at the structures of Remdesivir in complex with the RNA-dependent RNA polymerase, but more interesting is this picture in which it has been clearly demonstrated that Remdesivir is a covalent inhibitor, which is able to bind to the primer, blocking the elongation of the newly born RNA. In this way, Remdesivir is able to block the RNA-dependent RNA polymerase, to block a viral replication, and to stop the viral infection. But more intriguing is that, as hypothesized by our computational studies, that Remdesivir has a multi-target mechanism of action. In fact, binding to the primer, Remdesivir is also able to escape the proofreading mechanism mentioned before, which is, again, I would like to underline that the proofreading is one of the key mechanisms of the virus to preserve the viral replication, to maintain the viral replication, and particularly Remdesivir, blocking covalently the newly born RNA primer, is also able to block the proofreading, very likely inhibiting the exoribonuclease enzyme. However, the clinical trial outcomes with Remdesivir have been rather controversial. On April the 29th, the National Institute of Allergy and Infectious Diseases from the US announced that the treatment with Remdesivir was statistically much better than the treatment with placebo, reducing time to recovery for people hospitalized with advanced COVID-19 and lung involvement. In February and March, another clinical trial was completed in China, and Remdesivir was basically not effective in reducing the time for improvement from COVID-19. And more importantly, severe side effects were identified, required the investigators to terminate the trial. Again, I may speculate here that the problem number one was that Remdesivir should be utilized at the right point of the disease, at the right phase of the disease, because since remdesivir is a compound able to block the viral infection, if the hyperinflammation has already taken place, probably remdesivir has a much lower efficacy compared to its efficacy in the early phases of the disease. And finally, in the last few minutes of my presentation, I would like to mention about chloroquine and hydroxychloroquine, which are very, very old drugs. Chloroquine was discovered in 1934 as an antimalarial drug. Is also quite toxic, particularly because the half-life is remarkably long, up to a couple of months. Whereas hydroxychloroquine, the metabolite of chloroquine in this position is a bit safer relative to the parent compound. Both drugs have an interesting multi-target mechanism of action because they are able to block endocytosis. And blocking endocytosis allows chloroquine and hydroxychloroquine to block the immune response and to work as immunomodulator compounds. Indeed, hydroxychloroquine is mainly utilized in rheumatology, but the same mechanism of action is also able to block the endocytosis of the viral particles. This is mainly due, back to the structure of this compound, this is mainly due to the B-basic nature of chloroquine and hydroxychloroquine, which is responsible for the increase of the pH within the endosome, preventing the uncoating of, eventually preventing the uncoating of the virus. Interestingly enough, from our computational studies, hydroxychloroquine was also highly ranked as an inhibitor of the 3C-like protease. And actually, it has been reported afterwards in the literature that one of the possible mechanisms of action of hydroxychloroquine is also to block the viral protease. Let me skip this because it's conceptually pretty complex, and I'm at the end of my presentation. So, COVID-19 outbreak has challenged remarkably our health systems, but I would say also the ability of our scientific community to react promptly to emergency. And huge effort has been devoted to the discovery of therapies and drug repurposing in many different fashions, computationally or simply based on the clinical experience, has been the most obvious choice now. From the computational standpoint, traditional approaches, including pharmacophore structure-based methods, have been widely exploited. Also, machine learning, particularly in the context of natural language processing, has been quite interesting in finding new therapies for COVID-19. And using all this plethora of computational methods, we were eventually able to propose a combination therapy which includes an antiviral compound and an immunomodulator for treating patients in phase 2A of the disease. I cannot, for confidentiality reasons, tell you which are the two compounds that we are going to utilize in clinical trial, but in collaboration with hospitals, in the next few days, clinical trials will be launched using this combo therapy. Interestingly enough, if the trial will be successful, our combo therapy can be used at home, which is probably the next step for preventing the huge hospitalization that we experience in many European countries, particularly in northern Italy. I would like to thank my collaborators, to thank CICAM and you all for your kind attention.
Okay, thank you very much, Andrea, for this enlightening presentation. And as I mentioned at the beginning, we are happy to receive any questions through the chat channel that you have in YouTube. We will come back to these questions addressed to Andrea in the general discussion that we will hold at the end of this webinar. We will now stop for 10 minutes. So we will restart at 5 minutes before 4. And so hope to see you soon. Hello, everybody. Welcome back to the second part of today's webinar. We will start, as I had announced, by presentation by Modesto Orozco on the use of high-performance computing and big data in the understanding of COVID. And I remind you that you can keep on posting your questions through the chat or emailing them to the email address director@ccam.epfl, and Sarah Bonnell and myself, we will be collecting them for the subsequent Q&A session. So let's start. So Modesto, now the floor is yours.
Thank you. You see my screen, right?
Yes.
Well, thanks for the invitation. I'm a kind of CCAM addict, so I'm very happy to help to maintain the activity of CCAM talks in this dark period. So this is going to be not a pure scientific lecture as Andre's one. This is going to be a mixture of philosophy, history, and technology. And I will start by focusing what is happening. So is there anything new? So this pandemic is something new that has not happened before. And if you take a look to history, this has happened many, many, many times. Already in the Roman Empire, it was a big plague that kills 5 million people. The Black Death killed 200 million people, but from the Middle Age. So it kills 1/3 of the European population. More than half of the people in northern Italy and Germany. And more recently, just 100 years ago, the Spanish flu, that by the way was not from Spain, was from, it was imported to Western countries by American sailors stopping in China. Spanish flu killed a century ago between 40 and 50 million people. And if you think for a while, the way in which we stopped the epidemic has not, these ways have not changed a lot. So this is, you can see here, this was a cartoon from the Middle Age, not too different to what is happening now. And this is the quarantine strategies in the Spanish flu epidemic. And this is what we are doing now. So not big changes. And even the things that worry society at the time were not too different. So in Spain, we are now discussing when we are going to have football again. And this was the main concern in California during the Spanish flu, when they will have baseball again. And it was, apparently it even happened, a match, a baseball match with people masked. I don't know if we are going to see this, but from a historical perspective, our way to react against the pandemic has been the same than a century ago. Politicians have not improved much, society have not changed much. So are we going to have 40 to 50 million fatalities? And honestly, we don't have many indication that this is a less aggressive virus. If you look at the age pyramid, this is Spain, this is our life expectation. Our age pyramid now is worse than it was in the past. So this is the Spanish flu. This is the decay in life expectation because of the Spanish flu. Just as an average, this is the Spanish Civil War. So it was a dramatic pandemic. But the life expectation before the pandemic was a little bit 40. And now we are over 80. So we have an older population. So in principle, pathology can be even worse. And the world is more interconnected. We have more population, larger cities. Many reasons to don't be very optimistic, but honestly, nobody thinks that we are going to reach 40 to 50 million fatalities. I mean, we are now in the 200,000 range, maybe an underestimation, probably are more than has been recorded, but we don't expect 7-digit pathologies, or not even, of course, not 8-digit numbers. And the reasons are very clear, is new healthcare technologies and science. I'm not going to talk about this because I know nothing, But I'm going to talk a little bit on science, because now science is, at some point, has been accused to don't be reactive against the pathology with enough activity, enough results, and it's very important to be careful. So just before the pandemic started, there were a few hundred papers published every year on coronavirus. Now there are 600 papers published every day. And in most cases, I say papers, in reality are communications to our repository that have not passed any kind of peer reviewing. And what is bad is that our politicians are taking these unpublished results as if they were the holy truth, and make decisions based on papers that are retreat, that are not published at all, or in some cases, this is a problem in the US, but this is in Europe, where it was a big issue on the role of ibuprofen, that if you read the paper, and this is a good journal, if you read the paper, they never say that was a correlation. It was just an hypothesis. So need to be careful. And this is my last philosophical slide. I think we should take a very strong position as scientists, and there were many initiatives. This is from published from epidemiologists, published in Science, calling for transparency when they give numbers based on models of COVID spread. And this is a nice letter from almost 800 scientists in Britain to the government, asking, please don't press too much the scientific method. We need time, and we need to do things in the proper way. But if forget these problems that happen for this kind of rush to produce results very, very soon, the response of science has been very, very, very impressive. This is one of the first papers. And if you put numbers, in December, it was the first case that was confirmed in China. In December 31st, it was identified, a new virus. And just 5 days later, we had the sequence of the virus. And this is January. Today, we have more than 16,000 sequences of the virus. We know everything about how the virus evolved. By the end of January, we knew all the proteins of the virus. We had a good idea of what was the evolution of the virus. And when the pandemic was recognized as a problem, we already have an astonishing amount of information. So science has reacted really very quickly. And we know now the way in which the virus is made, the way how it invades the cell, the mechanism of replications, the receptor, the co-receptor, the proteinase that helps to put the virus inside. We know all the tools that the virus uses to be so good as an infector. And even as Andrea explained, we have already many or several of the key structures of the several key proteins of the virus. Typically, a structural biology project takes 1 to 5 years. You need to express the protein, cut crystals, or get good diffraction maps. It's a nightmare, and they managed to do it in 1 month. And the amount of information that this is providing is amazing. And it's amazing the amount of results that are collected both in the in silico level and in the lab by means of high throughput experiments that are giving rise. And we are talking on a couple of months to several drugs that are either in clinical use, was already talked about chloroquine, Remdesevir, and there are many, many others which are in clinical trials right now, not only small molecules, also antibodies. So the response of the scientific community has been, I would say, outstanding. But let me now focus in the field I know a little bit, that is computing. How computers are helping in fighting COVID. And computers are flops and bytes. So computers are machines that makes numerical operations very quickly, and are machines that help to store and to manage data. And we have learned a lot during this period on how the computers should be used in a proper way when there is an emergency like that one. So now all the supercomputer centers in the world have a priority, and COVID projects are, I would say, the first priority. They are the priority. So most of the HPC centers are not focused in doing simulation regarding to COVID. What we have learned is that the peak power of the computer is not the key element. So you can have a huge computer that actually is not as useful as a smaller one. The computer can be very fast, but then human setup of analysis can be the rate limiting step, can be the bottleneck. It doesn't make any sense to have an ultra fast computer that runs the simulation in 10 minutes if you then spend one day setting up the simulations and doing the analysis. We are running under pressure, so we need to be prepared because there is a lot of artifacts in the literature. The big reservoir for high performance computing time is still the multi-CPU computers. So big computers, as Marian Ostrom you have in the photo, with a lot of CPUs that run in parallel. Intra-calculation parallelism is crucial, but with the exception of a few calculations that are done in the US, with, for example, the antivirus, most of calculations people are doing now are medium-sized calculations. So it's not so tremendously important, the intra-parallelism as the inter-parallelism, because what people is doing are thousands of these simulations. And what we learn about the data, what the previous slide was about the flops, so the calculation, this is about the data. What we learn, we are generating incredibly large amounts of data. And this data is not useful if they stay in the place where it was obtained. Data must be shared and must be shareable in a fast way. And not only the data, the analysis must be accessible. It is not helpful at all to find a repository with a flat file with 10,000 trajectories. You don't know how to start. You need to have the data accessible to you, but you need to have the key analysis also ready to be accessible. It is very important, this is technical, I'm afraid, interoperability. So Andrea have shown the combination of molecular dynamics, accelerated molecular dynamics, docking, pharmacophore search. So if you do a simulation, you need to be sure that the results that you get at the end of the simulation can be used as an input for the new one. And you need to be sure that you follow some standards in such a way that people know exactly what you are doing and what are the expected limits of the accuracy in your calculation. And last but not least, we should be realistic. It's impossible to transfer the raw data. We are talking about exabytes of data. We cannot transfer this amount of data. It will collapse internet for ages. By the way, I must say that some of these problems we already anticipate, and I suggest you to take a look to this review we published a few months ago, actually explaining the problems of managing huge amount of data. And unfortunately, we found that all our predictions about the problems were real. But let's talk a little bit about the COVID problematics, the type of things that we are doing to make calculations more efficient and actually taking the most from high performance computing. And this is assuming a few things that for me are very clear. We have to do not a single calculation, we have to do many. The setup is quite repetitive. Calculations need to be done very quickly, so the results are very urgent, need to be done, and calculation that typically will take 2 weeks, you want now in 2 hours. And as I told several times, we need to do things in such a way that runs well in the current high-performance computing architectures. And these are some of the questions that actually interest us and for which we have developed what I'm going to show you. For example, how was the evolution? How was the steps that follow a virus that was happily in bat to arrive to human? And they were Globally, there were not many mutations, but locally in the infection point, in the AC2 protein, there were a large number of mutations. How this happened? What is the population dependence? How different populations with a different genotype react against the virus, against infection? And what is going to be the future evolution of the virus? The virus is mutating. You can connect and see every day the new mutations of the virus. There are many that has already been consolidated. How these mutations are going to affect the infectivity of the virus. These are the type of questions that interest me. And our approach is based on a type of, I would say is the gold standard right now, is a type of very painful calculation. Is named free energy calculations. It's based on rigorous statistical mechanics. And the idea is that using thermodynamic cycles, you compute the change that or the impact of a mutation in the binding. For example, this is the RBD. This is the part of the virus that interacts with the receptor in the cell. RBD is here. This is a very nice structure of a spike. RBD is here. We have high resolution structures for the RBD AC2. AC2 is the protein that acts as a receptor in the human membrane. And we have, Andrea has already showed this picture. We have a nice structure of the complex. So the idea, what the method allows us, is to compute what is going to be the change in the binding free energy when you mutate, for example, this position, you have, arginine, you mutate to a serine, and here you have an aspartic, and you mutate to an asparagine. So you can compute the change in binding, so the difference in these two numbers, not directly by computing mutation, free energy associated to the mutation, to the free energy change associated to the change from one residue to the other in the unbound state and in the bound state. It's a technology that is actually from the 90s, was developed by McCammon, Jorgensen, and others, and technically is a pain on the neck. And why is a pain on the neck? Because you need to compute this in a very long simulation, step by step. It takes 4 ages, and it is a type of simulation that doesn't take profit on supercomputers. And it's a pain on the neck because all the setup of the simulation is very, very costly. It's very low because you need to look at the best way to change one amino acid to another. So we have implemented a few ideas, and this is an idea that many people has been exploring. Instead of running equilibrium free energy calculations that are very slow, to go to non-equilibrium. And then there is a full physics supporting this, the Crook theorem, Jarzinski inequality, many others that say that, okay, something that you can do instead of doing this long simulation is to do many short simulations. And then what you compute, technically speaking, is not a reversible work, it's an irreversible work. But you can use statistical mechanics principle to demonstrate that actually if you compute the mutation from one direction and then you get this histogram of irreversible works, and then you compute from the other direction and then you get this other histogram, the free energy is going to be the middle point. It's the point where the two histograms intersect. And this has been proved many times to be quite precise. What is advantage? That then you can split the calculation very easily. You have 500, 1,000 independent calculations that can be launched in parallel to a computer. You don't need to be worried about intra-calculation parallelism because you have inter-calculation parallelism. Of course, you need to do that, but you need to do many things before. One thing that you need to do before is to automatize the input. So you need to have tools, clever tools that allow you to find the most favorable ways to transform one amino acid into the other. So to be less aggressive to the simulation as possible. And here we benefit of the work of Bert de Groot and all his team that have designed this PMX strategy that is very, very efficient to actually find reasonable ways to transform one residue into another. But you need to do more things on top of that. You need to automatize a series of tasks that are typically done by humans. That implies, set up, define the topologies, optimize, add waters, relax. So a lot of things that typically take a couple of days for an experienced researchers, and we have put all together. And we have had this to all the production run, and all this is sent as a single shot. And this is the work of Pau, Adam, Jose Luis. And once you have that, you have done this, you are able to actually run one calculation. But to be efficient, you need to run one calculation many times. So because all this workflow that we develop within the BioExcel consortia, all this workflow will run one mutation. But you want to explore, I don't know, 100 human polymorphisms against 400 mutants of the virus. So you need to launch many of these calculations. Here we were very fortunate to have the help of Rosa Badia and all her team at the Supercomputer Center, that they built a wonderful platform called PyComs that allow you to do that, allow you to make many calculations in a massive batch way, and then the platform will take care of running all together, assigning processor, detecting one is broken and need to be killed, and send another one. So it's really a fantastic tool. So this is an example of how the workflows are. For example, this is the setup workflow. And look at the numbers. For example, here, we can put in a single job 30,000 cores. When you have a supercomputer, you have many cores. And if you use only 100 cores, you are not really taking profit of the supercomputer. So with this technology, we can use 30,000. This is in the calibration, more than 15,000 in the production series. And actually, we can run simulation like this one. For example, I'm not going to talk about our science today, but this is an example of what I'm talking. This is a human polymorphism that happened in AC2. AC2 is this protein here. And this is the most common polymorphism in humans. So 3 of every 1,000 humans have this mutation, that is a lysine that changes into an arginine. And then you want to see what is the impact of the virus in this specific part of the population. And you can do it. You can do it by looking at the change in the binding, so the change in the infectivity of the virus because of the change. And I'm not going to bother you, but this is what we get, a small effect. So it's not negligible, but we expect that this mutation will make only slightly more susceptible the people to the infection. What is important for this technical talk, we did this in 3 hours. So in 3 hours, you can do this. This is what I mean by fast response. We are now have a technology that allows you to actually put HPC Center full steam to work in simulations, in COVID simulations. I'm going to jump now to the second part of my talk. I'm going to talk now about data. We are producing huge amount of data. We are able to produce terabytes of data every hour if we are running, for example, molecular dynamics. And this is a huge amount of data. It needs to be accessible. It needs to be fair. Fair means findable, accessible, interoperable, and reusable. So data is there and is there to be used. I talked already about curation, standards, metadata. This is what I mean by metadata. Your simulation to be useful, you need to have all this type of information. For example, what is the simulation conditions, the type of solvent, the protein, the type of analysis that was done, the force field, everything should be clear in the simulation to make it useful. And of course, the simulation need to be curated. And we are talking about thousands of simulations. It's impossible to do it manually. We have designed a few tools to actually curate, so check that the simulation is not rubbish, it's realistic. The data is there, and the data is useful. It is put in a database. If it's in a flat file, it's good if you know what you're looking for. But if you don't know what to do with the data, you need to have the data in a database. And to build a database, you need to have many things in mind. Data is not easy to move. So it's not trivial to move files in the petabyte scale. Data should have associated metadata to be useful, should be interoperable. The data must have associated analysis Otherwise, it's just dummy data. And then the analysis portals, which are very important, I will show you one in a second, should be very flexible because it should allow programmatic access and it should allow external people to incorporate the analysis that they want. So it is great. What would be great is to have a database and a data analysis portal where people can actually put their trajectories. And not only that, they can share the type of analysis that they are doing. It's at that time when we are going to be really useful in terms of our community. This is our technical approach. We have typically our databases, we have a server, we have a graphical interface, and inside there is a variety of database scheme. Typically now we are using Mongo quite a lot. This is the website and the database that we have built. It has around 100 trajectories right now. You can access, this is the production. We have one for development with a little bit more things, but this is the type of things that we can offer the community. You have the trajectories for many proteins, then you have information on the proteins, detail how the protein look like, even access to a specific points of the trajectory. Let me try if I can change to another screen to show you in real life how the server looks like, because this is going to be, I think, more useful. This is if you access, it's fully free. It's open to everybody that wants to use it and want to deposit trajectories. When you enter here, okay, you will find all the trajectories that are here. You can select, for example, this is not one of our simulation. It is from an American group. And then you have access to all the metadata that we have for the trajectory. You have many details. Now the trajectory is being released. You can modulate, this is very clear. This is the entire spike protein. And then you can see how the protein moves, and you can have access to many other tools regarding, all details on the protein. We can take a look, for example, let me check another case. I don't know, this one, for example. This is a complex. In this case, this is one of our trajectories of one of our projects. And you can see what the protein is doing. You see this very cool movement of the human. This is a protease, and this is a kind of opening and close movement of the protease. This is what is functionally relevant for this protein, but you can have many more data. I don't know, for example, you can see what is the main movement, the main principal component of the movement, this kind of rolling. You can take a look and see how the protein projects. For example, in this case, this is projection in two principal components. In this case, you can conclude that we have an equilibration problem because the trajectory is moving to somewhere else. Probably it was a receptor that was not corresponding to the viral protein, and then the protein is moving, it's drifting a little bit in one of the directions of the space. You can take detailed information, for example, on a specific snapshot. So you have access to the entire trajectory, you have meta-analysis, but you can have, for example, I don't know, this specific point here, which atom is that is oscillating a lot, and then you can get it. You see here that is not probably for your interest here, is not extremely relevant because it's far away from the interaction site. So you can do, well, but it's not far from the CD. Okay. You can take, for example, the data to be interoperable. For example, this is the radio generation, but for example, let's see that you are interested in cavities and you want to do docking, then you can scan through all the trajectories and find this specific point that is where the protein is more open and hopefully it's going to be a better target for a ligand. Then you can actually immediately take it and use this for a docking. So we can put here, and in our development server, we have many more analyses that can describe any kind of data you wish to visualize, and you can do it for entire series of proteins and actually share the type of analysis that you can do with other colleagues. So I think I'm finishing. Let me just share my presentation again. I told you we have in our database structure, we have access to individual snapshots, but you can combine individual snapshots for hundreds or thousands of simulations to get very robust data on a specific topic that can be of interest for researchers. And this is my last slide. So just thanking the people that has been involved and thanking you for your attention.
Okay, thank you very much, Modesto. And as I mentioned at the beginning, we will now have the possibility to discuss the questions that we have been receiving from all the people attending today's session. And if you have last minute questions, you can still keep on posting through the YouTube chat. So we can start. So Sarah, you can start sharing this part of the presentation.
Thank you, Ignacio, and thank you again to all our speakers and to the audience that has been following. Quite a lively chat also today. We have several questions for Andrea. I guess that going first had some advantages, but I would like to start from a comment that came in towards the end of Modesto's talk, because I think it's probably something where both of you can chip in. And that is, what is the relationship and the balance or the relative importance between high throughput calculations and high performance computing? I mean, and what is shifting the balance? Is this because we don't have good enough algorithms for these sort of problems, or is it because the nature of the problem favors high throughput? Modesto, maybe you can start.
Okay, yeah, okay. Yeah, I think that in general, in this type of cases, what we are looking at is at a high throughput type of calculation. So so of course there would be like one of the simulations we have in the database is 150 microseconds. It was done by D-Show, so it's a massive simulation. And I know Romy Amaro is simulating an entire virus, and there are very, very huge systems that are simulating. But in most cases, we are not, or most people are not facing now simulations of huge systems for a long period of time. They are working with, for example, analyzing many drugs interacting with the proteins, or analyzing many mutants of the protein, or analyzing many, many things. And then I think that the key point here is transform high-performance computer centers in a kind of machine that can tackle these problems, because HPC centers are good when you have a single calculation running for a long period of time. Now we have a different situation. We want to have very intensive calculations, doing many things, but getting the response in 2 hours. And this is what the type of middleware we have developed can be really useful, I think.
Okay, thank you. So now going back to Andrea and maybe to the very beginning of his talk, how reliable would you say is docking NMD to get results on inhibitor drugs binding? So do you have enough information to then move towards a therapeutic or an application stage? And then probably related to that, how large your ensemble of docking examples has to be before you consider it?
Yeah. Of course, usually the protocol should be much longer than what has been applied in emergency. For example, we plan in the very beginning to test our compounds in vitro. We have very good connection with at least 3 different labs with be as L3 level of security to use coronavirus, SARS-CoV-2 for testing drugs. But at the time of when our simulations were completed, all these 3 labs were closed down because of the lockdown. So the normal pipeline would consider also some co-crystallization experiments to have the experimental confirmation of the simulations before moving to the clinical and to patients, because in these cases we are moving directly to patients, not without any preclinical validation of the compounds. And so the lack of any experimental validation is something which is really missing in our normal pipeline, but the emergency called for that. We are now planning to purchase, as I mentioned before, there are at least 100 drugs that we were able to prioritize, and now we are going to simply purchase them. Now in Italy we are opening back, and we are going to purchase them and to send to viral laboratories for in vitro testing and to assess the potency of the compounds in blocking the viral replication. And concerning the ensemble, the answer is very similar. In normal condition, the larger the ensemble, the more accurate is the docking outcomes. So the ensemble could be created using many different crystal structures, for example, or you can run molecular dynamic simulations or enhanced sampling, MD, and now you can run multi-millisecond, multi-microsecond, or even millisecond long simulations. And in those simulations, you can generate a sample of conformation that, of course, can make all your outcomes more robust. In emergency, we had to run using very few, probably 2-3 different conformations, a bit more for the pharmacophore-based analysis, in which enhanced sampling allowed us to create probably close to 20 different conformations. And we use them for a kind of a small consensus.
Thank you. And then following up, we've heard quite a bit about the importance of free energy calculations, but maybe you could comment a bit more on the workflow. So the question was, if the drug fits into a target, so I guess if the docking is successful, then how do we know that the drug will really move to the docking place, and what is the relevance of the free energy barrier in all these calculations? Here, I think that both Modesto and Andrea can probably give some answers.
Yeah, Modesto, go first, if you want.
Yeah. So, yes, of course, free energy calculations are probably in principle, the most accurate tool that we have, to compute things. But they are really they are powerful, especially when you are playing with congeneric series, so molecules that are similar. So are very good for getting delta G's, more than to get absolute delta G's, but are expensive. So obviously you cannot screen through free energy calculations a library of 10 million compounds. This is why you typically focus, once you have good results in using docking or pharmacophore search, then you focus into the delta G's at the very latest stages, What I show you are big mutations that can be done in 1 hour, but this is under exceptional circumstances. Typically, a supercomputer center will be not very happy to allow you to have 15,000 processors to play with.
Yeah, the answer is very similar. Free energy calculations can be very accurate, even though absolute free energy calculations are still, at least in drug discovery, are still in a kind of infancy. Relative free energy calculations could be rather accurate nowadays, free energy perturbation or other chemical calculations approaches. Anyway, in this case, congeneric series are very difficult to be obtained. This is why we rather prefer to use binding kinetics, because binding kinetics is the compounds are different but relatively similar in terms of entropy, which is always very difficult to predict. You may try to assume that the K-off is a quite good surrogate, or even in some cases better than the KD. And by running unbinding simulations, which can be rather fast in the order of tens of nanoseconds, you may try to prioritize your compounds and check whether docking was working pretty well. We use ScaleMD, but other approaches historically I used also SteerMD, which was rather good anyway to rank different ligands binding into a binding pocket of the same target.
Maybe just a short follow-up, and then because Modesto mentioned this idea of using non-equilibrium statistical mechanics tools to also extract the same information. These are typically very noisy simulations, so it's a question about when, under which conditions it's competitive using this method rather than the standard equilibrium ones you were describing now.
Yeah, I mean, within BioExcel, it was one of our work cases, and we explore not for this specific system, for another one, obviously. And actually, in many cases, at the very end, you get the same result, but with less CPU cycles. So the point is that the non-equilibrium are extremely noisy, but what you play is with a histogram, and then the point of crossing the two histograms is quite robust. So you can we typically run in a standard equilibrium friendly calculation, you will run one simulation, right? In here, we ran 500, 1,000, and then you can see that from 500 to 1,000 or 200, you know, there is noise, of course, but there is not so much noise. And of course, a lot depends on the way in which you make the mutations. So the topology change, but I think Bert has done a great job on this. The big advantage really is, however, not that it's faster, but the point is that it fits better to supercomputers. So supercomputers are very bad. If you can put in a single calculation no more than, I don't know, 256 processors to be efficient in terms of parallelism. And then you need to use these 256 and then wait and another window 266 and so on. In this way, you can use 266 in parallel for 500 times if you have power enough. This is the big advantage, is that it's better fitted to supercomputer architectures.
But what would you say is the limiting factor now in accuracy in these predictions, and how far are we from being useful? I mean, if we get things with error bars of, I don't know.
I mean, in the case that we have seen, for example, we have explored, for example, the mutations from the bite virus to the human virus. And this is about 21 mutations. So it's a dramatic change. And there is a lot of charge mutation. This is quite noisy. In this case, you need to burn quite a lot of CPU time. And at the end of the day, you don't have more than, I would say, 2 kcal error. But in the other mutations, for example, in human polymorphism or SNPs related to the virus, I would say that the accuracy, if the force is right, the accuracy is in the order of point something kcal. So, because you repeat many times and you get many times the same answer, different snapshots, so it seems to be quite robust.
So Andrea, maybe a few more specific questions on your talk. Can you comment on the recent findings on apparent inefficacy of hydroxychloroquine?
Chloroquine. Again, there are many, as I tried to mention during my presentation, there are many different trials with the same drugs and sometimes controversial outcomes. This can be due to many different reasons, to many different hypotheses. For example, from our experience collaborating with hospitals here in Italy, but also in Germany and also in Spain, initially hydroxychloroquine was used at suboptimal doses, which were the doses of malaria, of rheumatology, and actually for triggering any visible, any appreciable antiviral activity, the loading dose should be much higher. And then after a few weeks of trials, physicians understood that the loading doses should have been at least the double of the doses used for malaria, and then the drug start to be a little bit more active. I would say if it is used in the right phase of the disease, it can help in between 50 and 60% of the cases. I don't know which is the opinion of Modeste on that, but I'm seeing that there is a very, very strange evolution of the disease worldwide, which can be, and also the therapies associated with the disease are rather different and can have different effects on different people. And what really we are missing, my personal point of view, but again, I would like to listen Modesto's opinion, is that we are missing genomics data worldwide to try to understand which is the impact of the viral infection and the impact of the different therapies in different subpopulations. For example, in Italy, we have almost 3 times lethality in northern Italy compared to the southern Italy. 3 times, which is statistically relevant. It's not 1.1, 1.2. And also different therapies have quite different outcomes in different niches of population. And there is no possibility to make any comparison because simply we are missing data. We are missing genomics and also transcriptomics data to try to understand which could be the impact of the virus on different populations. And it is something that we should consider now after this very, very dramatic outbreak.
This is a very, very good point. I think everybody will agree with Andrea. So it's, I mean, we have 16,000 virus sequence, but we have very, very little information about the patient. And I think it's absolutely stupid because, I mean, now my institute is doing PCRs for detecting the pathology, and in the same tube, you have the genetic material of the person, of the patient. So it would be, technically speaking, straightforward to actually have the virus and the person on the same Excel file. And this type of questions that are absolutely shocking. For example, the very, very low prevalence in kids. I mean, it's almost impossible that why, So, of course, a lot of things will be related to the immune system, but we need this genomic data. In principle, the only, at least in my institute, the tools are likely to be here, but the only problem is a problem of, is a bureaucratic problem. So you need consent from the patient because, and things are done in such a rush that we are forgetting things that are very obvious. So I think if we have 16,000 virus sequence, we should have 16,000 genome sequence. Compare one by one, and then with the clinical record to see if this guy has gone to phase 1, that is, I don't know, a firefighter that was for any reason checked, or is actually in very bad shape in the hospital. If we don't have this information, we are missing, a big part of the story. That's absolutely clear, Andrea.
And building on this idea of sharing information, although in a different context. So the idea of drug repositioning is intuitive and direct, and many groups around the world are working on this. Is there a database or repository with the results of experiments and over the different targets? We've heard, I think, a little bit about that in the Escalate for CoV project last as maybe a first attempt, but...
No, as far as I know, there has been no a real collective activity in the field of drug repurposing. And it is something more that we are missing, actually.
Okay, so back to maybe a more specific question. Wouldn't targeting the proofreading enzyme favor mutation of the virus and help it adapt to treatment? Andrea, this question was originally aimed at you, but you seem to be frozen at the moment, so maybe Orozco can step in.
Yes, Andrea is frozen. I just see black. Yeah, I mean, it has been quite a lot of controversy, really, whether, you know, the virus, this virus changes not much. So I think it has 0.3%, something like that. I have notes around. It has a low mutation rate compared to other viruses. So it has mutated a lot because it has been infecting millions of people. But it doesn't have a tremendous mutation rate compared to HIV. So I work with HIV, and HIV is incredible. It generates tons and tons of mutations on the same protein. In this case, the mutation rate is small, and it has an advantage and a disadvantage. So the advantage for us is that we cannot anticipate a huge mutation, like happens with HIV, for example. So we can, if we design a good drug, we cannot expect the virus reacting, generating 50 type of mutations that will inactivate the drug, right? This is good. But on the other hand, it's bad because the proofreading protease warranties that a high efficiency in replication of the virus. And this is what Andrea actually suggests. So if you lose this fidelity in the replication, what it means that when the virus replicate, it will generate, I don't know, 100 copies, and of these 100 copies, 90 will be wrong, will have lethal changes. And then the total infectivity of the virus will be lower. So you have these two phases. On one hand, you decrease the robustness of the virus if you remove the proofreading domain, but on the other hand, you have a less predictable virus. So Andrea, you're back.
Yes.
So I was answering your question.
Sorry for that. I don't know what's happening, but
no problem. That's the beauty of a live broadcast, I think. Ignacio, do we have more questions, you think?
I think we have gone through many of the questions, I would say, but maybe it's related to this question about different virus and having more general strategies. There is the question about focusing, how about targeting the human proteins that are targeted by the virus proteins as a more good strategy for drug repurposing. Again, that's probably more for Andrea initially.
Yeah, we should consider many different possibilities. For example, now the hyperimmune plasma, which is an historical antiviral approach, has been quite successfully in many hospitals in Europe. And this is a general strategy. This is a general antiviral strategy, but of course cannot be used in the very early steps of the outbreak because you need patients who recovered, and then you need the blood from these patients. You need to extract plasma to make it hyperimmune and then to re-inject into patients. Another important breakthrough has been the identification of the different phases of the disease. For example, treating patients with antiviral compounds when they are incubated in the very last severe steps of the COVID-19 is basically useless. Useless probably is too strong, but it's not very helpful. And in some cases, you even worsen the clinical condition of patients because these antiviral compounds have some quite strong side effects. That they are used for Ebola, for HIV, are not very easy drugs, I would say. And so the identification of the different phases and the immediate repositioning of drugs for the late steps where the hyperinflammation took over the viral load has been probably one of the major breakthrough, which came first in Italy, unfortunately, because we were the first, and then was spread throughout the rest of Europe. We didn't learn much from the Chinese because we thought that was only there and stayed there like SARS-CoV-1. Yeah. And actually, this was not the case this time, unfortunately.
Yeah. Okay, I mean, there are more questions, but I think we already had quite a lively discussions with many topics that have come out beyond your presentation. So I think we should thank again Modesto and Andrea for your time and for making this very insightful session. And I want to tell you that these presentations will be staying in the YouTube channel, and also through the CCAM website, we have a devoted page where we store today's session and all the previous ones. And I also invite you, I hope to see you again next Tuesday, where we will hold the last session of this series around COVID and computation. So thank you very much and stay safe. See you next week.
Thank you. Bye-bye.